Hello everyone and welcome to VIGYAN MARG. We are very excited to start our brand new PYQ series for GATE Biotechnology and Life Sciences called Bio Booster series. In this series, we will go through previous year's GATE questions, discussing each and every topic in detail. Our goal is to not only help you to solve PYQs but also to build your strong concepts, which will be useful for other competitive exams like CSIR NET, DBT, ICMR, JRF, etc. And this series is going to be very important for your exam point of view. And I am Arnav, a student of IIT Guwahati, and I will be guiding you throughout this series. And before starting, make sure to subscribe this channel, Vigyan Marg, to get more videos like this. Today we are starting with GATE 2025 paper. So let's get started with our first question. So in the first section, you will find the general aptitude part, which we're going to discuss later on in a separate video. So for today, we are starting with the biology section. So in the first question, said that Koch's postulate was established by Robert Koch while working on a disease caused by. It's a very simple and basic question, and the answer for this question is Bacillus anthracis. So as I have said that I will discuss each and every topic in details. So here I will discuss a little bit about the Koch postulate, which discovered by the Robert Koch, and we all know that Robert Koch is one of the founder of medical microbiology, and he formulated Koch postulates, a set of four principles that link a specific microorganism to a specific disease. So if we discuss about Koch's postulate, in the first section, he said that the microorganisms must be present in all cases of the disease but absent from a healthy organisms. Okay, in a disease organisms, the microorganisms will present, and in a healthy organisms, microorganisms will not be present. In the next section, he said that the microorganisms must be isolated and grown in a pure culture, and from the disease organisms, the microorganisms be isolated and grown in a pure culture. Okay, and in the third postulate, he said that the cultured organisms should cause the same disease when introduced into a healthy host. And these cultured organisms, when introduced to a healthy host, will cause the same disease. Okay, and in the fourth postulate, he said that the microorganisms must again be re-isolated from the experimentally infected host and identified as being identical to the origin. And when the same microorganisms will be isolated, and these features will similar to the same microorganisms which was isolated very earlier. So this was the Koch postulates. And next question: Corynebacterium diphtheria causes diphtheria in human only when this bacterium is infected by. So Corynebacterium diphtheria itself is not always pathogenic. Okay, not always pathogenic. The toxin responsible for diphtheria, which is called diphtheria toxin. Please keep a pen and paper with you to make notes, which will going to help you further. And the toxin is encoded by tox gene. So this gene is not a part of the bacterial chromosome. This gene is not a part of the bacterial chromosome, and instead it carried by a lysogenic bacteriophage. It carried by a lysogenic bacteriophage. Okay. Specifically, the beta phage integrate into the bacterial genome and bring along with the tox gene. Like the beta-phage infect the bacteria and which it brings the tox gene which will further integrate in the bacterial cell. This phenomenon is an example of lysogenic conversion when the bacteriophage imparts new traits like toxin production it to its host bacterium. So the phage bacteria, the phage beta is responsible for this, which will help the Corynebacterium diphtheria to cause diphtheria in human. And moving on to the next question. So here YT, a bacterial population whose growth is given by this equation, and where lambda is the growth rate constant, y01, y14, and the value of the lambda is. So it's a very simple equation. If we write the equation, we should take a separate page for that. Okay. So the equation given like dy by dt equal to lambda y plus 2. So further, if we do like dy by y plus 2 equal to lambda dt, and if we do both sides, we took ln, so ln equal to ln into y2 lambda t plus c. This is the constant. Here we can get it's like e to the power yt plus c. Okay. From here, this equation we are getting, and very initial step what is said that y1 and y1 equals to 4. So from that equation, if y0 equal to 1, here y0 means t equal to 0. So from that equation we will get what we will get if we put the value of y is 1, 1 plus 2 equal to e to the power y into 0 plus to the plus c. Here we will get 3 equal to, we know e to the power 0 equal to 1, so plus c. So e to the power c comes 3, so we will put this e to the power c equal to 3 in this equation. So from here we will get yt equal to 3 e to the power yt. Okay, and the next step, if we have asked that y1 equal to 4, so here we will simply put this equation here. Minus 2, there is y plus 2. So here we will get 4 equal to 3 into e to the power y1 minus 2. 3 e to the power y into 6 and e to the power y equal to 2. You can get y equal to, both sides we will take, so the answer will be y is equal to ln 2. This is not plus, this is into. So the answer will be ln 2. Okay. And moving on to the next question. The minimum value of the function fx is equal to x plus 4 by x. And for this x greater than 0. And first we will do the derivation of this equation. F of x is equal to 1 minus 4 by x square. Okay. So here if we take f of x is equal to 0, then what we will get? 0 equal to 1 minus 4 by x square. So here we will get x square is equal to 4, x is equal to 2. And further, if we put the x is equal to 2 in that equation, what we will get? F of 2 equal to 2 plus 4 by 2. 2 plus 2 is equal to 4. And here 4 will be the answer. Moving on to the next question. The diversity in T cell receptor is generated by, the T cell receptor, which is called TCRs, and we also know about the B cell receptors. Both show high diversity, but the mechanisms are different. And BCRs, we know this. And the first option is said gene rearrangement, somatic hypermutation of rearranged V and V region, gene conversion, and class switching. So in the case of gene rearrangement, which is generally known as VDGA recombination, the TCR diversity is mainly created by the VDGA recombination during the T cell development in the thymus. And V we know is the variable region, and D is the diversity, and J is the joining segment of the recombinant randomly. Additionally, the junctional diversity, insertion and deletion by the nucleotide further increase the variation. This is the separate part. And if we move on to the somatic hypermutation, which happens only in B cell. This happens only B cells after the antigen exposure, introduces point mutation in rearranged V region to increase the antibody affinity, and does not occur in T cell. This does not occur in T cell. And if we talk about the gene conversion, another mechanism in some species for antibody diversification, not involved in TCR diversity in human. And class switching is a different portion involved in B cell changing antibody isotope like IgM to IgG or IgA. This is called class switching. So not relevant to TCR, T cell receptor. So our answer will be the gene rearrangement, which is called VDJ recombination. This is our answer, gene rearrangement. And if we move on to our next question, which one of the following is true for piRNA? And piRNA is known as PV interacting RNA. Okay, and this piRNA is small non-coding RNAs like 24 to 34 nucleotide long, and they interact with the PV protein. They interact with PV protein, and their main role is silencing transposable element like transposon, line, sign, etc. in germline cell, thereby protecting genome integrity. So this mainly responsible for silencing transposable elements like line, sign, and retrotransposons. And one thing you have to remember that it is will not interact with P element. So here the options is given piRNA silence responsible for element in germ cell. piRNA is abbreviation of P element interacting RNA. piRNA modified 2' OH of the ribose in methyl group. piRNA is long-term non-coding RNA. So here we can get the it's helped in the answer. Option A will be correct answer. Pi silence the responsible element of germ cell. So moving on to the next question. Which one of the following coenzyme is utilized by alanine racemase for the conversion of L-alanine to D-alanine? Alanine racemase catalyzes the interconversion interconversion of L-alanine to D-alanine. So L-alanine to the alanine racemase will help enzyme will help the interconversion of L-alanine to D-alanine. Okay, and the further this reaction is essential in bacteria because D-alanine is required for peptidoglycan synthesis in the cell wall. Okay, and the alanine racemase is uses in pyridoxal phosphate as a coenzyme, and in fact this pyridoxal phosphate forms a chief base with a amino acid, and it stabilizes the carbanion intermediates, allows the alpha hydrogen to be abstracted and replaced on the opposite side and leading to racemization. Okay, so for here the pyridoxal phosphate will be the correct answer. And if we talk about the other option, thiamine pyrophosphate, which involved in decarboxylation reaction, decarboxylation, and tetrahydrofolate is one carbon transfer reaction, and flavin mononucleotide is a redox reaction. Okay, so the next question, correctly match the following monosaccharides with their respective epimers. So it's a very simple question. So first we will see about the D-galactose. D-galactose is the C4 epimer of D-glucose. Okay, D-galactose is the C4 epimer of D-glucose. And we see about the D-mannose. D-mannose is C2 epimer. D-mannose is a C2 epimer of D-glucose. And now D-allose. So if we take up the option P4 and R2 is correct. So here we can say that option A will be correct. So here Q will be 1. D-allose is C3 epimer of D-glucose and D-tallose is C4 epimer of D-mannose. Okay. Just you need to look at the option. The next question. Correctly match the following product classes with their representative product. So biofuel, we know butanol is a uh, biofuel produced by fermentation. So for that, P option will be P3. You can see that P3 is there in three options. And next we go to bioplastic. Bioplastic polylactic acid is a standard bioplastic polymer. It's a very simple Q4. Q4. And again, cellulase is an industrially used enzyme in biofuels, textiles, and more. So industrial enzymes will be cellulase. So R1. And cephalosporin is a prominent antibiotic compound. We all know that. It's very simple. S2. And the option D will be correct answer here. The next question. Which one of the following host is used in mammalian cell culture for production of glycosylated recombinant therapeutic protein? And this Vichia pastoris is an yeast expression system. Okay. And SF9 is an insect cell line, and acidic oil is a bacterial system, and Chinese hamster ovary is used for mammalian host system, provide a proper protein folding, disulfide bond formation, and human like glycosylation. That's why this is FDA approved glycosylated therapeutic protein are produced from CHO cell. Okay, so the answer will be option D, Chinese hamster ovary cell. The next question. Which of the following features are used in distinguish archaea from bacteria? So in the first option is gram staining. Gram staining is based on cell wall thickness and the structure, not a fundamental distinction. Some archaea may stain gram positive or gram negative, but this is not reliable to separate archaea and bacteria. So gram staining will not be the proper option. And peptidoglycan, next option is peptidoglycan in the cell wall. So bacteria usually have peptidoglycan in their cell walls, and archaea lack true peptidoglycan. Instead of, they may have pseudopeptidoglycan proteins or polysaccharides. So on the basis of the peptidoglycan in the cell wall, we can distinguish between the archaea and bacteria. And present of N-acetylglutamine NAG may be present both bacteria and archaeal cell wall. 16 is RNA sequence. 16 is RNA sequence. Archaea were the first distinguished as a separated domain by Carl Lewis based on the unique 16 is RNA. So Carl Lewis. We all are familiar with this name. And this is the most fundamental molecular creation for distinguishing archaea from bacteria. So option D will be the correct. This is an MSQ, okay? This is an MSQ question. You need to take care of these things. So both option A and D will be the correct answer here. So on the basis of the peptidoglycan and 16S RNA sequencing, we can distinguish the archaea and the bacteria. Again, moving on to the next question. Which of the following enzymes are involved in the biogenesis of the mRNA? So again, this is also an MSQ questions. Okay, is an R involved in biogenesis of mRNA. So if we talk about the function specifically, if we talk about the mRNA biogenesis pathway. Okay, so first the transcription happen and transcribe as a pre-mRNA which is primary mRNA in the nucleus. So the pre-mRNA in the nucleus is there, and there is the dorsa. Dorsa is there process the pre-mRNA into process this pre-mRNA into pre-mRNA. Okay, this is the primary. This is one is the primary mRNA, and this one is the pre-mRNA. Okay, and this pre-mRNA is still in the nucleus. Okay, this pre-mRNA is still in the nucleus, and this pre-mRNA is now exported to cytoplasm. And here there is another element is present which is called dicer. And dicer is another RNase 3 enzyme which cleaves the which cleaves this pre-mRNA, okay, which cleaves this pre-mRNA like 20 to 23 nucleotide double stranded mRNA. So there is a we get this mRNA, and this from this the one strand is loaded into the RISC complex which is a RNA induced silencing complex for the gene silencing. Okay, and this mRNA is further helped in gene silencing. So here the we are getting this dorsa and dicer from this option. It's very two important thing, dorsa and dicer. And you need to remember dorsa function in nucleus and dicer function in cytoplasm. Dorsa help to cleave the primary mRNA, and then dicer help to pre-mRNA to mature mRNA, and further this mature mRNA by the help of RISC complex help in RNA silencing. Okay, so Cas9, if you talk about the other option, Cas9 is used in CRISPR-Cas system. In CRISPR-Cas system, and XRCC4 is a DNA repair protein. Okay, so you will read it in DNA replication. So our here from we get the A and D option is the correct option. And moving on to the next question. Which of the following separation process are based on molecular size? This is again is MSQ question. So we all know about size exclusion chromatography is also called gel filtration chromatography. Separated molecule based on their hydrodynamic size, large molecule elute faster, okay, and a smaller molecule enter the pores of the stationary phase and elute later. We all know this, okay. And ion exchange chromatography, ion exchange chromatography based on the charge, okay. This is one is the size. And membrane filtration use semi-permeable membrane to separate the molecules according to their molecular weight cutoff. Smaller molecule pass through and larger molecule is written. So this is also based on their size. And ultracentrifugation is like separate based on the size, shape, density, all these things included, not purely on molecular size. For macromolecule like protein, sedimentation depend on both the size and density. So this one is partially correct. We can say this is also based on the molecular size. So here we are getting A, C, and D is the correct answer. Okay. Moving on to the next question. Which of the following shows optical activity and 100 millimolar concentration in water? So NaCl, solution of NaCl is an ionic compound. It has no chiral center, so it is optically inactive. Okay. In solution of D-glucose, D-glucose is a chiral. D-glucose is a chiral molecule and exists as a enantiomers also. So in solution, it rotate plane polarized light. So it can rotate plane polarized light. So it is optically active. Solution of glycine. Solution of glycine is simplest amino acid you all know. It's a alpha carbon is not chiral. So therefore it is optically inactive. And we talk about the L-proline. L-proline has a chiral alpha carbon, so it rotates the plane polarized light and is optically active. So option B and D here is a correct answer. And this is an MSQ question. So moving on to the next question. Which of the following fluid exhibit non-Newtonian behavior at 25 degrees centigrade? Okay, so first option is toothpaste. Toothpaste is a pseudoplastic type things and its viscosity decrease with the increasing CR rate. So it's a non-Newtonian fluid. And if we talk about the mercury, mercury is a metallic liquid. Viscosity is constant regardless of CR rate. So it's a Newtonian fluid. And brine is a salt solution. So a simple aqueous NaCl solution behaves like a water. It's a Newtonian fluid. So this option is also incorrect. And blood plasma. Blood plasma is exhibit CR thinning behavior due to protein like fibrinogen. It's a non-Newtonian fluid, although less pronounced than whole blood. So it's a non-Newtonian fluid. So option A and D will be the correct answer. Now moving on to the next question. Which of the following compound have the same degree of reduction per carbon molecule? So it's a very easy question. We'll discuss this very easily. Okay, so degree of reduction per carbon molecule. For that, we have a formula. Degree of reduction per carbon. Please note this formula in your copy. So gamma is equal to 4 carbon plus hydrogen minus 2 oxygen minus 3 nitrogen. Okay, so this is a very important formula for this question. So what is given here? Glucose. Glucose, we know the formula C6H12O6. So if we put it here, C6H12O6. So here we can get 4 into 6 plus 12 minus 2 into 6 minus, there is no nitrogen, 0. Here is 24. We will get 24. And another formula we need to remember, degree of reduction is equal to the total we get, okay, we divided by carbon number. Here the carbon number is 6, okay. So if we get that degree of reduction 24 by 6, we get 4, okay. So this is the first, and second one is lactic acid. Lactic acid, the formula is C3H6O3. C3H6O3. So from here we will get 4 into 3 plus 6 minus 2 into 3. So from here the 12 plus 6, 12. So here the degree of reduction will be 12 by 3. We get 4. Okay. In the third option, we said acetic acid. So the formula of acetic acid is C2H4O2. From here we will get 4 into 2 plus 4 minus 2 into 2. Here we will get plus 4 and minus 4 is 8. So here we will get degree of reduction by it is 4. Talk about the fourth option, formic acid. Formic acid, the formula of formic acid is CH2O2. From here we will get 4 into 1 plus 2 minus 2 into 2. Here we can get 4 plus 2 minus 4 is equal to 2. The degree of reduction is 2 by 1. Okay. So the question is, following compound have the same degree of reduction per carbon molecule. So we get that option 1, 2 and 3 we got the same degree of reduction, which is 4. So here our option A, B and C will be correct option. So this one is a two question. So you need to remember this formula, okay? So if we're moving on to the next question. A recombinant protein is secreted extracellularly in soluble form by E. coli culture. Which of the following downstream processes are involved in the purification of the extracellular secreted protein? So first option is said that cell disruption. So here is said that the secreted extracellularly soluble form of E. coli. So the protein is already in extracellular, so we do not need to break open the cell. So cell disruption will not be the correct answer. Next, membrane ultrafiltration, and it can be used to concentrate and partially purify the extracellular protein, can remove small molecules and concentrate the targeted protein. So membrane filtration, ultrafiltration will be the right answer. And next option is solubilization of inclusion bodies. Inclusion bodies occur with insoluble intracellular protein. Here the protein is soluble and extracellular, so no solubilization is required here. Okay, so solubilization of increased protein, no need to do that. And again is liquid chromatography, essential for further purification like ion exchange chromatography, affinity and size chromatography. To further purification, we need liquid chromatography. So here option B and option D is the correct option. Moving on to the next question. If the doubling time of a bacterial population is 3 hours, then its average specific growth rate during this period is. So we know the equation between doubling time and specific growth rate. Know that doubling time is known as TD and specific growth rate is mu. We know the equation like mu equal to ln2 by TD. Okay, so here the TD is given 3 hour. So we just need to find out the mu. We know the ln2 6, 5, the answer will be or so the answer is 0.231. Okay, so this is the answer. It's very simple. Just need to remember the question. For a mechanically reversible isobaric process taking place in a closed system involving 5 moles of an ideal gas, the temperature increases from an initial value of 300 Kelvin to a final value of 450 Kelvin. If the specific heat capacity at constant volume Cv is given as 12.5 joule per mole per Kelvin, the gas constant is 8.314 joule per Kelvin mole per Kelvin, and the amount of heat transferred through the system will be. Okay, for that the process is mechanically reversible isobaric process. Okay, so the system is closed system with 5 mole of ideal gas. So here two different temperature is given. Okay, extra page for that. Here T1 is given, T2 is given with 50 Kelvin. Okay, and the R is 8.314, CV and CV is 12.5. So we'll simply write down these things. 5 and the R value is 8.314 joule per mole per Kelvin. Okay. So if we calculate the change of temperature is 450 minus 300 Kelvin, which is 150 Kelvin. So if we calculate the ice very fast, it is said that this is an isobaric process. Okay, and for the isobaric process, the formula is, we know the C formula of Cp. So Q is equal to Ncp delta T. Okay, N is the number of moles and Cp is the specific heat constant at pressure, and it is the temperature change. It is the temperature change. Okay. So here the Cp is given, but Cp is not given. And here it also said that this is an ideal gas. Ideal gas. And for the ideal gas, Cp is equal to Cp plus R. Okay, from here you can calculate that Cp, which is Cp, which is given 12.5, 12.5 in, and R is given 8.314. From that we will get 20.814 joule per mole per Kelvin. Okay, here we have found out the equation. So here we simply put the equation here. It has the heat transfer of the system. So simply put these things in this equation equal to n. Previously I just said that n is equal to 5 because the 5 mole ideal gas constant. N is equal to 5 and the Cp is 0.814 and the temperature change is 150. So from here we will get 15610.82. Okay, so the answer will be 15610.5. You can round off the nearest integer is 1.6 and 1. So the answer. Now moving on to the next question. The allele associated with the albinism in human is recessive. The probability that an albino male and a carrier female will have an offspring with normal skin pigment is. Here we have said that albinism is a human recessive disorder. So, like in a Punnett square, if we cross it, so, no, capital C, small c, and small c. So here we'll get small c, small c, small c, capital C, small c, small c, small c. So here we're getting that out of 4, so here is a albinism is a recessive disorder. So out of 4, 50%, you can see 50% is recessive, so which will cause the disease which will cause a disease. And here the question asked the probability of that one albino male and carrier female will have an offspring with the minus skip treatment. So here we can get that 4. So we need to find out the percentages. So here 2 out of 4 will be infected. So here his chance is 0.5%. Okay. Now moving on to the next question. The contrast length of pDNA molecule that encodes a bacterial protein of 33 kilodalton is as nanometer. Consider the average molecular weight of an amino acid is 110 dalton. The helix rise per the base pair is 0.34 nanometer. So we know that 33 kilodalton is dalton. So from here, the weight of the amino acid is given is 110 molecule. So we'll easily got the number of amino acid from here into amino acids. So now each amino acid coded by 3 nucleotide. Okay, so the number of nucleotide would be 900. So 900 nucleotide is the coding stand, and DNA is double stranded, but the contrary length depend on the number of base pairs. So we use the number of nucleotide in one strand like 900, which correspond to 900 by 2 450 base pair. It's not basically right. Okay, so if we make it simple, each codon, we know that each codon has 3 nucleotide. Okay, each codon has 3 nucleotide. So 300 codons will be 3000 codon will be 3 into 3000 into 3 is 900 nucleotide is on one strand corresponding to the 9. So one strand will be 900 nucleotide, and another strand there will be similar 900 base pairs will be there. So if we want to calculate the contour length of the DNA, so if we take only one strand, okay, one strand we will take. If we take the one strand, then here it said that rise per base pair is 0.3 nanometer. So here if we do 900 into 0.34, we will get 306 nanometer. So here the answer will be 306 nanometer. I think you get understand. In one strand there is 900 base pair and 900 nucleotide, and another strand there is a 900 nucleotide. So to calculate the length of the DNA, we only took one strand, so we can get the length of the DNA similar to the other strand. Okay, moving on to the next question. Within the Michaelis Menten framework, the ratio of V0 by Vmax when the A is equal to 20 into Km. So we know the first we will write the formula Michaelis constant Vmax is by Km plus S. So here we have asked for the V0 by Vmax. So we will simply write V0 by Vmax equal to S by M plus X. So here A is equal to 20 into Km. So 20 Km by km plus 20 km. So 20 km by 21 km. So if we calculate this, we will get something 0.952. So if we round it into two decimal place, we can write 0.95. So this will be the right answer. Moving on to the next question. Consider the nonlinear algebraic equation e to the power x minus 2 equal 0 using the Newton-Raphson's method with the initial case of x not is equal to 1, the approximate value of the root of the equation after 1 iteration. So if we recall the formula of Newton-Raphson formula, we will get so Newton-Raphson formula said that so here the function is f of x e to the power x minus 2. Okay, it is given here. So if we derive this formula, we get f of x is equal to e to the power x. So if we take initial case is initial case x0 is equal to 1. So from that, if we take initial x0 is equal to 1, here we will get x1 is equal to x0 minus f of x0 by dash x0. So x0 is 1, and here we will write now we need to calculate the values of this u1 and 2 by calculating this we get the value like a4x0 by dash. This is equal to 0.264. Now x1, this one is for the 1 minus is there, so x1 will be 1 minus 0.264, 0.76. Okay, from here we will get the two decimal, we will get 0.7, answer will be 0.7. Let's need to the put the formula by replacing these things. This equation e to the power x minus 2. Moving on to the next question. The value of k for which the linear equation 2x plus 3y is equal to 6 and 4x plus 6y is equal to 3k have at least one solution is 2x plus 3y is equal to 6. The equation is given 2x plus 3y equal to 6 and 4x plus 6y 4x plus 6y is equal to 3k. So you can easily do that things like first we will multiply it by 2, so we will get 4x plus 6y is equal to 12, and downside we will get 4x plus 6y is equal to 3k. Minus minus and minus, this option is for cancel out, and here we will get 3k is equal to 12, k is equal to 4. Okay, here the answer will be k is equal to 4 times 4. So we'll end here and we will continue the next portion in the second part of this video. And till here, if you like this video, please like, share and subscribe. And if you have any doubt, you can comment it down. I will reply definitely. And if you want to get this PDF, you can join the Telegram channel VIGYAN MARG, which was pinned in the description box. So thank you for watching. Please make sure to subscribe this channel. And till then, bye bye. See you in the next video.